Welcome everyone. My name is Jim Anderson. I'm faculty in agronomy and plant genetics. And this is the first of our Forever Green initiative meetings for the semester. Don Weiss is on the call. He's usually chairing these events, but I wanted to introduce Katherine for this one. Special welcome to all of the I Prefer collaborators. So this is the nationwide CAP grant that's led by Western Illinois. Minnesota is a very prominent participant in that grant. So you folks have the invite for the semester, so you can tune in to any other topics that you may be interested in. So we have folks on, I guess, mostly from Illinois, maybe a few from Missouri. So I would like to introduce Katherine, and I think probably everybody on the line here is aware that she will be leaving us soon. She has taken a tenure track position at the University of Nebraska. I'll let her talk more about the details of that position. But next week is her last week at the University of Minnesota. And so she's been with us for 5.5 years. So she came in July of 2015. So Pennycress had just been harvested, and she was taking over kind of a fledgling program, and she's really built a strong foundation for the future. I think most of you know that she's kind of the hub for all Pennycress activities at the University of Minnesota. She's been our spokesperson. She's been the person that connects everything, the agronomist, the breeding program that she directs, the genomics team with Ratan and David Marks, pathology with Senyu Chen and Cody, food science with Pam Ismail, probably leaving some people out, and all of the national collaborations as well. So she's kind of leaving a hole for us, but she's got a nice share drive. All of her files are getting dumped in there, and I've already been asking her for opinions on what we should be directing our program to in the future. As a little bit of a stopgap measure, we have hired Ratan Chopra to partially fill some of Katherine's breeding responsibilities. So Ratan had been with David Marks for a number of years, so it's sort of a lateral move. He's still going to be working out of David's lab, but officially he's in the Department of Agronomy and Plant Genetics as of December 21st, 2020. So as we go forward, we'll probably get through this growing season and then decide how we want to replace Katherine's full responsibilities if we go for a postdoc or more technician help for the program going forward. But rest assured, I think we're in good hands through this growing season. So I guess, well, one last thing that I want to about Katherine is, so she's actually research assistant professor. So this is a non-tenure track position. She's been in that position for a little less than a year. And I went back and reviewed the letter that I prepared to have the dean sign off on that. And it's really amazing the number of grants that she's authored. So there's a NIFA $1 million grant, there's an MDA grant supporting breeding activities, numerous Forever Green Initiative grants, a couple of Walton Foundation grants, and then she was obviously a key contributor from Minnesota on the I Prefer CAP grant as well. So, you know, she's single-handedly responsible for several $1 million worth of Pennycress support. So whoever replaces her is going to have to keep the money flowing for this program. So with that, Katherine, go ahead and share your slides and tell us about your next adventures and the state of Pennycress.
Yeah. All right. So I'm going to hide these video panels. So I'm going to talk for a little bit today about the about kind of Well, it's really kind of a retrospective look at what I've been doing, as well as things I'm excited about going forward. And then Zenith is going to share an update on his PhD research, which was funded through that NIFA grant. So, hang on. There we go. So, like Jim said, my last day at the University of Minnesota is February 1, and I will be leaving the Pennycress and Camelina program behind, and I am headed back to Nebraska. I did my PhD in the small grains breeding program at the University of Nebraska. I worked on nitrogen use efficient hard winter wheat, which is one of the reasons I was so excited to go into the Pennycress program, because I could really continue that breeding for nitrogen use efficiency in the system, and use my skills in plant breeding to make it easier for farmers to have more sustainable cropping systems. And so I'm really excited to share with you all that I accepted this position at Nebraska right before Christmas, and it's actually, I will be replacing my PhD advisor, Steve Benzinger, as the new small grains breeder. So I will be working on hard winter wheat, winter barley, and winter triticale, and I'm really excited to see where that goes. So with that, my objectives while I was here at Minnesota were to really develop a breeding pipeline for pennycress improvement and domestication. When I started, we had a graduate student, Kayla Altendorf, who had done some initial germplasm screens on some wild pennycress collections. And it was really my job to take that initial data and turn it into a breeding program. And so that's why I did a lot of grant writing, got a lot of research funded and a lot of equipment funded. I've worked to identify genetic variation present in the U of M pennycress collection. You all, if you're familiar with pennycress, know that there is phenotypic variation, but it's really hard to see it out in the field. It doesn't appear to the naked eye as much. So we really wanted to understand what genetic variation is present. And I have also worked on helping to identify and validate those value-added traits for pennycress commercialization that our genomics team has worked on. So just a quick reminder, if you're new to Forever Green meetings, that pennycress is a brassica winter oilseed, and we are basically totally domesticating it from scratch. It's a wild, weedy species that's been naturalized all over the world, but it's pretty high yielding and high oil content. So even though the wild type oil is not edible, it is suitable for industrial purposes. And part of the U of M and the whole I Prefer team's goal is to eventually move beyond that industrial use for pennycress oil and get it to that food grade higher value market. So here's a little bit of a timeline of the U of M pennycress breeding and genetics program. And y'all remember that it started in 2013. The Marks Lab focused on the genomics aspects with Kevin Dorn, as well as started that mutagenesis population that has been so important for establishing some of these domestication traits and high value end use traits. Jim's lab focused on breeding and working with the wild variation present from pennycress collections. And Kayla made the very first pennycress crosses that I'll be talking a lot about the progeny from those in 2013 and 2014. When I arrived in 2015, that's when we made our first data-driven crosses. And from there, it's just really been about expanding our team, working with more people, more traits, more aspects of pennycress breeding and domestication. I work have really enjoyed working really closely with David and Kevin Dorn and now Ratan. Also, the agronomy team, Russ and Scotty. I mean, most of you probably have heard that Scotty and I are on the phone like about once a day, occasionally more when necessary. So we've really developed this close collaboration because when we're developing these new crops, it is important to think about how these crops are going to roll out on the landscape as we're breeding them, trying to do all of this work simultaneously. We work with Pam Ismail in food science and now with the Plant Protein Innovation Center. We have a really important project looking at SCN resistance and susceptibility in pennycress with Dr. Senyu Chen from plant pathology. I don't have Catherine Bushley's name on here, but we also have started doing a lot of SCN work with her in PMB. We also have this really awesome relationship with Covercress. I've really enjoyed working closely with them and starting to test some of their lines up here in Minnesota while they test some of our lines down in Illinois and Missouri. And that information that we share, observations that we see, has really made both of our programs better. Starting in 2019, we added some work with our Forever Green commercialization team, Connie and Colin. They are absolutely great to work with. And I'm always asking them, hey, what traits do we actually need in pennycress? And that's an ongoing discussion. Our team has expanded with I Prefer and now the I Prep program. So it's really been cool as part of these last 5 years to see how the pennycress community is expanding. So we're finally getting to the point we're starting to increase our Pennycress 2.0. So that is a edible, a reduced shatter line. We're working on so many different traits that will eventually be in Pennycress 3.0, 4.0, and I see this continuing on. So the way our program has worked in the Anderson lab, since we're focusing on that wild genetic variation, we start with a cross and we're just inbreeding and advancing generations, making selections as we go. And there are some challenges with this in that our wild pennycress germplasm tends to have a lot of seed dormancy and a lot of seed longevity, which is not good when you're running a breeding program because we can't reuse fields like we would in a soybean program or a wheat program. And so that means we have to really think creatively about how much space we use each year because we have to keep in mind that we need to have somewhere to put our improved lines in the future where they won't be contaminated with our wilder, less domesticated lines. But really, one of the benefits of pennycress is that it has a short life cycle, so we can take advantage of that. Over the years, we have multiple crossing blocks for during the year when we can. COVID kind of messed that up last year, but it tends to be 20 to 40 crosses per block. A lot of wild by wild, now wild by mutant crosses to get those important traits into the breeding program. And now we're selecting some elite lines, we're really focusing more on forward breeding. Those crosses are then advanced in the greenhouse through a single seed descent modified method. And if we're talking about mutant crosses, we would do some marker-assisted selection. Currently, most of our lines go into the field in a single row as F3s. Ideally, I'd really like to get that to F4s in the future. It's just been a little challenging. If we have more technical support or a greenhouse technician, we could definitely get that advancing faster. But these single rows, we grow them just in St. Paul, and we really strongly select for emergence. So how well and how quickly do they come up out of the field? And when do they flower? After that, the next year, the selections go to an unreplicated small plot nursery. Again, these are small plots because we're trying to save space on campus. This depends on the number of families we have in those single rows, but it tends to range between 80 to 120 lines. Again, we're still selecting for emergence, for early maturity, as well as this is our first look at seed yield and oil profile. From there, we advance to a 2-rep multi-location small plot trial. This tends to be around 36 lines, and we've moved to planting this really at 2 locations. We used to do a few more, but we tended to lose them, and just figuring out how to harvest was challenging. So we usually plant this at St. Paul and Rosemount. You can see up here in the corner. This is where we really start to select for seed yield as well as oil yield, and looking at performance across environments. Anything advanced out of that AYT1 goes to a 3-rep multi-location trial, still small spot plots, but we have added in Morris for some years. For 2021, due to COVID restrictions, we just planted this in 2 locations again. Here we start to select for performance across locations as well as years. Pennycress is very plastic, and so we see a lot of environmental and year effects. And so lines might stay in this trial for a couple years before finally we advance them to because I'm not that creative with trial names, I just call the big AYT. And this is a 4-rep multi-location. It is not a small plot trial. It is a larger plot trial. Got a typo there. And we have been planting this at 4 locations in Minnesota: Morris, St. Paul, Rosemount, and Waseca. So we get some pretty good environmental data here. And because we are planting these at a higher seeding rate, more akin to what Scotty's planting in the agronomy trials, and these are in larger plots, we get a better idea of how the lines are performing in an agronomic sense. And so that's really how the program has functioned. I have a question mark here because right now we don't really know how long lines are going to stay in this trial. We don't necessarily have a variety release outlet yet because these lines don't have the mutant traits in them yet, because we were doing this breeding simultaneously with the mutant trait development. So that's something that my successor will be figuring out. So here's a look at some of the lines that have been in the 2 small plot AYT trials over the last few years. This is a subset of the lines because it's hard to display all of them. So you can see in the corner that these are what our small plots look like. These are actually our 2021 trials here on St. Paul campus. So if we look at this graph, the first 2 lines here are the wild unimproved Czech varieties that I use. You're all familiar with Min 106. That is the basis of the mutation breeding population, as well as it's been our agronomy trial lines for the last probably about 5 years. Ames 29509 is a wild accession from Grin, and this was one of the highest yielding lines in Kayla's master's thesis trials. And so we've used that as our higher performing check. And then all of these lines here are from crosses that Kayla made when she was here. And they've been selected for mostly emergence, earliness, as well as yield. So you can see that we're really looking for them to get over that Min 106 bar. You can also tell that, yep, we have quite a bit of variation from year to year. So it makes it really hard to make good selections, but I think we're getting a better handle on that as we get more testing years. And so really these are what I want you to take away from this is that we are making improvements over these wild lines, and we have some lines to be excited about. This is the results of our, and I see that I missed some of my animations here. So I'll explain what these mean in just a little bit. This is our big plot advanced yield trial results from 2020. And we unfortunately lost our Waseca site in 2020. It was planted late and it was just cold and wet in fall 2019, and the plants didn't emerge, and we ended up plowing that one under. You can see that Morse is a pretty challenging location. Most of these plants did emerge in the spring, which explains the much lower yields. Rosemount tends to be a pretty high yielding site, and then St. Paul is almost always our highest yielding site. It's a little bit warmer here, and because we're on campus, we tend to get that location planted first. So that's some of the differences between the locations. Again, we have the same 2 wild type checks. Next, this is a trial where we collaborate with Ratan and David to test some of the improved Min106 mutant lines. So this first one has an edible oil phenotype. And then this next one is edible oil plus reduced shatter. And what's really exciting is we see that we are making some pretty good yield gains across all the locations by having that reduced shatter trait in there. And then here's some of the top breeding lines. These do not have any traits, any of these mutant traits included yet. So these are some lines that we've been testing. Again, we want to see that they're out yielding Min106, ideally across all locations. So that's what that was. And so then these 2 lines, they were very promising early on. And so we added them to our crossing block. And this actually, we have ROD1, Phe1, so these are edible oil. We have low glucosinolates, an early flowering trait, and reduced shatter, all in a SIB line that this Min16 167 has just recently edged out. So we have those all in a SIN line. Our Min16 207 was a pretty good performer for several years, and we've got 3 of those traits in. We will be working with Ratan and David to get other traits in. And then these 2 lines that I have starred here, 16172 has been a line that we've started testing with CoverCress as well as our I Prefer trials. And that's been performing very well across our the I Prefer testing sites. And so we will start adding those mutant traits into this line. And then 17077 is a new line that I'm actually really excited about. It's one of the most uniform lines I've seen. It's a little bit shorter than Min106, so it doesn't tend to lodge like Min106 does, but it's also quite a bit earlier. And that's why these yields are actually lower, because this line matured and started shattering before any of these other lines were matured. So I'm really excited to see what this line could do once we get that reduced shatter trait in. And here's just a closer look at that line. This is at flowering, so you can see it's pretty much in full flower here, and Min106 is just starting to flower. You can also see that we've made progress on breeding for good emergence with these lines. So you can tell that here, and then also in, these are these same 2 plots in a drone image. So you can see that Min106, I think this line is our Ames 29509. But most of our breeding lines, here's 17077, I think this is 16172 here. So we've really made a lot of progress in getting that better emergence. So hopefully lower dormancy, more uniform germination. So I'm really excited about that. I also am very excited about what we're doing with the I Prefer variety trials. This is become a regional test trial for Pennycress breeding programs. There's not very many of us, but it's good to get this program started now. So we have entries from U of M, Illinois State, Western Illinois with WIN, and then CoverCress. Whoops. Last year, the 2019 to 2020 field season, we had this planted at 8 locations and we're able to harvest 6. And the figure on the right-hand side is our results from the Rosemount site. We also had it planted at Morris, but the Morris yields were quite low, so I don't have those in the chart. And you can see that we do use some different regional checks here. And what was just exciting to me is we do see that our breeding lines, as well as this new TT8 mutant from John, are really outperforming some of these older regional checks that we're using. So this wasn't a really high yielding trial. You can see the mean was pretty low. And it just goes back to, we did have kind of a tough fall in 2019 with late plantings. I'm really excited to see how these trials do in 2021 because we had a much better fall planting in Minnesota. It was dry, but both Rosemount and St. Paul and even Morris and Waseca look pretty good. So we ended up with some well-timed rains. And this is an overall summary of how the I Prefer Variety trial worked across all of our collaborators. So you can see WIN's trial here in Macomb, Normal, the 2 Minnesota sites. Oh, sorry, that's supposed to be on the end. Didn't check that. R does some like funny things if you don't specify that you want to keep your order. Then here's CoverCress site. You can see the benefit of, they have a lot more growing degree days than we do. So Pennycress is definitely well suited to the I states, and then our collaborators in Ohio. I do want to point out again, John's TT8 line tended to top the trial pretty much everywhere. So that's pretty exciting to see, as well as our Minnesota lines. Here's 16172 in the blue, tends to do pretty well across locations. So I'd say overall that we are making good progress, and that's what I wanted to share about our yield trials. The last thing that I really wanted to share is a project that Ratan and I have been working on for the last couple years, and that's developing a new Pennycress genome. We have had a draft genome, thanks to Kevin Dorn's work, since 2014, and the challenge is that this genome was very fragmented, and it was making it hard for us to both identify new traits, or the causative genes for new traits in the mutation breeding program, as well as do efficient back crossing. So we decided to, with funding from the Minnesota Department of Agriculture, embark in developing a new genome, and we are nearly finished. We hope to have that out by the end of the year. And this is basically, we've dropped our scaffold number significantly. Our largest scaffold size has increased, and the total coverage of the genome has also increased pretty significantly. The Pennycress genome is about 530, and we've got about 500 covered here. I have this starred because just in the last couple weeks, we identified a problem with these long scaffold we were calling chromosome 1. So we've identified a misassembly there, but we think we've figured out what happened, and we'll be hopefully moving forward and fixing that very quickly here. As part of this genome project, we decided to do some resequencing analysis of 39 Pennycress accessions. These were both spring and winter annuals because we really wanted to look at the diversity within pennycress. We used collections from across the US, Canada, Europe, the Caucasus region, so that's Armenia, as well as a line that Wynne had collected or received from Tibet. And we've really selected these lines based off of some initial genetic screening work that I had done using GBS. But for this, we wanted to use whole genome sequencing and really take a much deeper look at the genetic variation of these 39 lines. So this is just a look at the results of that whole genome sequencing work. We found about 14 million variants. Most of those were SNPs. And however, we do have almost 900,000 insertions and a little over 1 million deletions. So we do see this variation across the whole genome. Most of the work that we've done so far has been focused on working with the SNP data, but Anthony Brusa has just started helping us analyze some of this indel data. So we're really excited to see what kind of structural variation we see in some of those pennycress accessions. For the first time, we were able to look at LD decay across the Pennycress genome. And what we found is that it really decays by about 100 kbs. Here's our halfway point. And this is really a pretty typical result for most self-pollinated crops. So no major surprises here, but this was good to see, and this will help us with our with Zenith's upcoming GWAS analysis, as well as continuing to understand how we can use markers to make decisions for pennycress breeding.
Katherine, tell folks that don't know what LD decay is.
So it's a linkage disequilibrium, and basically we're looking at, imagine, so we have a bunch of markers here that are really close together, and how we're looking at how that linkage or do these markers segregate together across wider and wider distances. And so what we would expect is that markers that are very closely linked or are close together in the genome are going to be tightly linked, and markers that are farther apart will be not very closely linked. And so we want to see this decay happen at a relatively small distance. Otherwise, it can make some of the genetic analyses that we do a little bit more difficult.
Thank you.
Yep. All right, so however, we really we can't do a lot of our analysis with a panel of around 12 million SNPs. It's just not computationally possible, as well as it just kind of dilutes the information. So all of the downstream analysis that I've been doing so far, I've been completed on a panel of about 200,000 SNPs. This panel was generated based on filtering for minor allele frequency. So we want to make sure that any markers in this panel are not the same base pair throughout. So we need to have variation. We don't want to have very much missing data, and also this is that linkage disequilibrium. We want to have markers spaced evenly throughout the genome, basically. Here's our principal components analysis for these 39 accessions, and what was really exciting to me is that it does match pretty closely our GBS analysis. So we have a high degree of confidence in the work that we've already done. we see that our Armenian lines are incredibly diverse compared to all of our other lines. Our European spring lines tend to be right about here, but other than that separate group, our spring and winter lines tend to be very intermixed. They do not form separate populations. We do have a separate group here that is lines from more I would say colder climates. This is a lot of the northern Minnesota collections as well as those from Canada. And so we also have started looking at population structure with this whole genome sequencing data. And again, it was very comforting to me to see that we found 4 populations using this Ivano method. So the peak occurs at the most likely number of true populations. And so for us, that is, we could divide those 39 accessions into 4 distinct groups. And for the most part, these groups really match our GBS data. And so we have those a very distinct Armenian population with not much mixing through the other populations. We have our northern climate group. You can see that there starts to be some more mixture between the different populations in some of these lines. And then our warmer climate group. There was some differences between our GBS analysis and the whole genome sequencing analysis, and that's resulted in, we still have 4 groups, but one of them is this new group that in the GBS data, these 3 lines were mixed between these 2 populations. So with this higher density SNP, we can get a little bit more of a, not necessarily a different story, but maybe a clearer story. So we haven't really gotten into what makes these different compared to our GBS analysis. So that's a an area for further investigation. With our GBS data, it actually divided the Armenian groups into 2 different populations, while our whole genome sequencing data puts them together. So that's some interesting observations that I don't really have an explanation for, and we will continue working on that. So with that, I want to wrap up. I have a pretty long list here, but I know it's not extensive, so I apologize if I have forgotten your name, and I really didn't even put our I prefer collaborators on here. So thank you all. I've really enjoyed working with you for these last few years. And I've appreciated learning from all of you, and I hope you've all learned from me too. So with that, I'm going to stop sharing, and why don't Zenith, you go ahead and share, and if people have any specific questions for me, you can go ahead and put them in the chat and I'll answer them while Zenith is talking.
I'll give a brief introduction for Zenith. So Zenith is a PhD candidate in my lab. He's funded by the NIFA grant that Katherine wrote. So that the objectives of that grant is to look at seed size and oil content in pennycress. Zenith is using a mapping approach that he will describe. David Marks's lab with Ratan, they're using a mutational approach, and John Sedbrook is the other co-PI, and he's using a CRISPR approach for those 2 traits. And Zenith, good news about Zenith, he just passed his oral prelim exams. What was that, a week or maybe 2 weeks ago? So he is a true PhD candidate. So go ahead, Zenith.
Thank you, Jim, for the introduction. My name is Zenith Pendekar, and as Jim said, I'm a PhD candidate with Jim at the University of Minnesota. And today I will be talking to you all about the research I've been doing for the past couple of years in Jim's lab. Let's see. Okay, so we all know that pennycress is an exciting oilseed crop, and there's a lot of descriptions of like the progress that have been made by Ratan and the Marx Lab in the previous semester's presentations, and then again with Katherine today. But I would like to highlight one point where like there is still a need to increase seed size and oil content in Pennycress to maximize its commercialization potential. Like Katherine said, we are currently in trying to grow out the Pennycress 2.0, but like we would really like to be able to introduce some of these traits like bigger seed size and higher oil content in our future pipeline, like Pennycress 3.0 maybe. And why we really want this is so that we can have better handling ability for the conventional combines that our farmers like to use. And we also want to improve tolerance for variable planting depths and improve yield and productivity for better commercialization potential. Now, this brings me to the overview of my dissertation chapters. What I'm really trying to do is understand the genetic control of seed size and oil content in Pennycress. And for the purpose of this meeting, I'll be talking to you all about the mapping aspects of my project, where one of the aspects is the association mapping of seed size and oil content in diverse germplasm, while the other aspect is the QTL mapping of seed size and oil content in two biparental RIL populations. Now, for the association mapping panel, we have a diverse germplasm collection from the University of Minnesota and our collaborators. And what we have done with it is we have gone ahead and sequenced it with GBS and gotten some high quality SNP markers associated with our diversity germplasm. And we've also planted this germplasm at 4 locations over 3 years. And what we really want to do with it is we want to phenotype for seed size and oil content over the diverse germplasm and perform genome-wide association studies. What we want to do is like find associated QTL and identify candidate genes and of course report findings from our research. The other aspect of the mapping work we're doing is in biparental mapping, where we have created 2 biparental RIL populations where they have the progress is that we finished creating them in the summer of 2020, and now they've been planted in St. Paul in the fall. So we'll get to phenotype them this summer, and we are also looking to sequence all the individuals and parents this spring. So we'll have all of the necessary phenotype and genotype data to perform QTL mapping and find associated QTL for these important traits. Now, to go into our association mapping panel, as we can see, we have a diversity panel collected from a wide area, but most of our germplasm still comes from North America. There's some from Europe and Eastern Europe, like the Caucasus regions that Katherine talked about, one from Tibet, and a couple others from South America. Now, what do we do with this? We went ahead and grew our entire germplasm, and we generated genotypic data for the diversity of germplasm. We, we isolated DNA and performed GBS. We have all of our data generated from the newest version of the genome. So we have up-to-date SNP calls with the latest version of the genome that we'll be using for our association mapping work that we're conducting. So just looking at the general data that we have created, and going back to Katherine's presentation, we know that our Armenian population is the most genetically distant, like most diverse line that we have. And it's always good to see that our results are validated through different methods. Like Katherine mentioned earlier, there's these yellow lines are the Western European lines, the spring lines that we're talking about, and all of the other lines from North America and Canada are mostly interspersed with each other. Now, along with the genotypic data for association mapping, we also need to create good phenotypic data from the diversity germplasm. So for year 1, we focused on the seed size and oil content related traits that the grant is based on. And then for seed size, we have the 1,000 seed weight, seed area, seed length, and seed width, classic seed size related traits. And for seed composition, we're mostly focused on oil content, but we also have data for proteinuric acid, oleic acid, linoleic acid, and everything else that the NIR gives to us. In the year 2 and year 3 for the same germplasm, we have gone ahead and we are going to get all of the same data, but we have also added like flowering time, plant height, seeds per pod, and average pod size into the mix to get a more complete picture of what is present in our diversity germplasm. Now, as I said, we are going to be doing a lot of phenotyping, but how are we going to do it? So we are lucky at the University of Minnesota that we have access to these two machines. The Pertin NIR gives us high throughput non-destructive phenotyping capabilities for seeds at a very fast rate, and we don't have to lose any seeds in the process, which is really good. And for seed size, we have this Marvin seed analyzer in the Anderson lab where, as you can see in the picture here, the seed bed actually opens up where you can just pour your seeds and it automatically calculates all of your seed size related traits, and it's very quick. So we have pretty robust data from these two high throughput phenotyping machines that we are comfortable moving forward with our analysis. For the new traits that I introduced in year 2 and year 3, what we wanted to do was we wanted to also look at average pod size and seed number per pod for the diversity germplasm. And this is just an example where we take around 10 seeds from the middle of the plant and we scan them and use a software called ImageJ to basically get a quick idea about the average area of the 10 pods for the line. We then go ahead and calculate the, not calculate, but count the number of seeds in each of the pods, and we calculate the average seeds per pod per line. So this is a project I'm working on with an undergrad, and she is focusing her undergraduate thesis on it. So this is very exciting for me to be helping new students with this. Now, just to look at the phenotypic variation present in the seed composition traits in Pennycress in the association mapping panel, we can clearly see we have good phenotypic variation for oil and protein, erucic acid, as we expect. We know that Pennycress is pretty high in oil, and as we can see, it's shown here with like around 30 to 36, 37% in the field. In the greenhouses, it's a little bit more, but since we're focused on the field data, this is more accurate. And then again, we also want to see the phenotypic variation for seed size in 3 different environments. So what we can clearly see here is, as Katherine mentioned in her presentation earlier, there is definitely an effect of different environments on Pennycress. And the leftmost one here is from Rosemount of last year. And Rosemount was really dry, and our plants were much smaller compared to St. Paul in both of the years. So as we can see, the average seed size for Rosemount is a lot smaller than for St. Paul in for both years. So we definitely have to keep that in mind, that environment plays a big role in some of these quantitative traits that we are working with. Now, just to go ahead with showing the preliminary data on what we're looking for, our main traits of interest is this TGW is 1,000 grain weight, where we are trying to look for genomic reasons associated with 1,000 grain weight and oil content associated with Pennycress germplasm. Now, as we can see, we do have some pretty robust hits, significant QTL for both of these traits in our germplasm, but this is just for one population. This is just for one location. We still need to perform a combined analysis for all of the locations, and I'll probably be presenting a similar kind of data in a following meeting for Evergreen. So this kind of wraps up the association mapping portion of my talk, and I'll move forward with the biparental mapping. For the biparental mapping, we have recombinant inbred lines that were developed with crosses that were made by Katherine again. So I inherited those crosses when I came into the lab, and I progressed them to the generations, and we derived F6 derived F7 RILs that have been planted in St. Paul field as of last fall. And then what this cartoon picture is basically trying to show us is that we want to find genomic reasons that are associated with either oil content or seed size in these RIL populations. Now, to just talk about the current status of the QTL mapping populations, we have as I mentioned earlier, completed the generation of these populations. Then they have been planted in St. Paul. We also grew a few plants in the greenhouse for seed increase, as well as to give us enough source for DNA to get the genotyping going as well. And we will have them phenotyped in the summer, this phenotype this summer, and we'll be performing QTL mapping and sharing our results in a future meeting. Now, when we say crosses were made between genetically divergent lines, what I mean is the parents are very different to each other genetically as well as phenotypically. So if you can see, if you look at the principal component analysis on the left side here in the graph, the Armenian line and the Min106, the Minnesota line, they are very genetically distant, as shown in this PCA. And the progeny from that cross makes up the range between the two. And as we can see, the average seed size for Min106 is around 0.95 grams per 1,000 seeds, whereas for the Armenian lines, it's around 30% more. So there is definitely some variability there that we can exploit. Now, as I said earlier, we grew a few lines in the greenhouse, I mean in the growth chamber, to give us like a preliminary idea of how the lines, the RILs were performing for the traits of interest. And as we can see here, the two lines that the two crosses that we have in the RIL generations, even with the growth chamber data, we can see that they do so pretty good distribution. The left one is the Armenian by Min106 cross, whereas the right one is Min77 times Min77 crossed with Ames 31501, which is mostly North American lines crossed to each other. I mean, this same thing with the oil content, where we do see some distribution, good distribution. And I'm only showing a few traits because for at least for seed size, as we can see from these figures, the seed size related traits are very highly correlated to each other. So the seed size difference in 1,000 grain weight between the two parents basically shows that they're different in length, width, and area as well. And it's always good to see the classical trait relations like oil and protein have inverse correlations between, with all of our with our calculations. So that was encouraging to see, and we'll be very interested to see more robust data coming out of the field this summer. For some things that I would just like to end with here is like when I was taking some germination and vigor notes on our field this past fall, one thing I noticed was our field looks extremely very, very good this year compared to the last years. We had pretty early planting. The weather looked amazing. Everything was really nice. But what I did notice while I was making these measurements when I was walking the fields was one side of the field was significantly different from the other. So what I basically mean is, even though I could see good germination throughout the field, I could see that the plants were less vigorous on one end of the field than the other. So what I wanted to do was to see whether or not this was, actually could be visualized with just numbers and spatial modeling. And what we clearly see is that one end of the field looks amazing, while the other end is very, the blue aspect in the fitted spatial train basically shows that the other end of the field is struggling for whatever reason. Maybe very compact soil, maybe just lack of nutrients in that part of the soil. I don't know yet. This is very new data that I need to discuss with my advisor and figure out what is going on. But this is one of the things that points to us that we need to account some spatial variation in, even though our fields are small, we need to account for spatial variation in our fields to basically move ahead with some of these projects, because we would need very accurate measurements to be finding QTLs for both of these traits. And that is then my presentation. And I'd like to acknowledge my advisory committee with Jim, David, Aaron, and Katherine, who have been a great help to me throughout. And Ratan, who has also been amazing to work with, answers all my questions. And we get a lot of things done. So that is it. Thank you, everybody, and I'll be taking any questions.
Thanks, Zenith. Cody, I'm just looking at the chat. I don't see any questions there. Anybody's welcome to unmute and ask questions.
Zenith, Ratan here. What was the correlation between uric acid and oil content in your population?
Correlation between uric acid and oil. Let me just check while I'm talking with you here.
Yeah, because I mean, both I think University of Minnesota, Illinois State, and even CoverCrest has seen that zero uric acid lines tend to have lower oil content. So I'm wondering if you saw a positive correlation in your population.
Let me go ahead and check it here. Give me 1 second. I'm just opening a separate presentation. There was actually no correlation. There's like 0.04. So very weak positive correlation.
Okay.
So Katherine, I had a question for either Zenith or Katherine. I know as we move farther north, I know a lot of work from Scotty and Russ are looking at relaying crops and double cropping and things like that. And we've talked about Minnesota. Under that situation, you wanted to breed a taller variety to get up and over the primary crop. But you were talking about shorter varieties. So I was wondering if you could comment on that. Do you see a difference, Zenith, in plant height and the pod sizes and things like that as you alter plant height?
So I'll start. We want, it's a little bit, when I made the comment, I can't remember if I said this as well, but it's shorter than Min 106, and Min 106 tends to lodge quite a lot. So that's one of the things I like about that line, is that it's shorter than Min 106. It is not, it should not be too short for the relay cropping system. And one of the challenges with Pennycress is that yield and plant height, I have a really nice figure and I didn't include it today, but they are very positively correlated. So the taller plants tend to yield more, and that's one of the reasons why I find the Min 17077 exciting, is that it yields well even though it's shorter. So we'll see if that holds, because that line is in the I Prefer trial this year. So we'll see how well that holds up. This last year, that was the first year it was in the big plot trial. So that was where we had first observed that.
Yeah, in relation to that, in our association mapping panel in the diversity germplasm, we do have like a few lines that are extremely tall, but they are also extremely robust. So they do have bigger seeds, bigger seed pods. They are taller, but they're also like very robust to not lodging. So there's a couple of lines that are really, that look really good, but most of the times in the diversity germplasm, Pennycress does decide to lodge if it's taller than Min 106.
Thank you.
No problem.
Katherine, could you say some more things about 17077? So in terms of the parents of that, was one of the parents early flowering? Is it more early flowering or just?
So off the top, so that is a 3-way cross. And it was, so I can't remember the 2 parents of the original cross, but one of them is the line Don collected in New York, which had been one of the higher yielding lines in the trial, or that Don and Kevin Betts evaluated before they really settled on Min 106. So that it was a single cross topped with min 106. So it's basically 50% min 106, which is why I find it exciting that it's earlier and that it's a little bit shorter and less prone to lodging. Off the top of my head, I can't tell you. I don't remember either of the other parents being significantly earlier. We could probably use Zenith's data to double check. But this may just be one of those happy flukes that you find in plant breeding.
Thanks.
This is Daniel. I have a question. So what is about the heritability of the seed size?
What's that question for me? The heritability for seed size, as we can see in our population, is around like 0.5.
So how far I see the yield for the best from the I prefer is about 650 pounds per acre for the TTA. That is the best one. So what is the yield, the minimum yield that can be commercialized, and how far away are we from that minimum yield to be commercialized?
So, I mean, that's an interesting question. I know CoverCress has used 1,500 pounds as a guideline, and it's going to be challenging for us to consistently meet that bar right now in Minnesota, especially with the environmental plasticity of pennycress and the, you know, when we have challenging environments, it's just not going to yield that much. I think we have some lines that are really pretty good in the pipeline, and I also think, you know, that yield bar level is going to change as we add some of these other traits in, and as we think about what systems we're putting this in. And that's something that Connie and Colin have been very good about helping us figure out. We also have quite a few different grad students who have worked on different economic analyses that Connie and Colin have been utilizing for some trying to figure out how will farmers make money with pennycress and camelina. So
since I come from a commercial soybean breeder, soybean breeding history is interesting for the seed size and seed number. Before, we always trying to breed for the larger, the better. Mm-hmm. And then maybe 7 or 8 years ago, the soybean seed began to sell by seed count. And the company by sell larger seed soybean, they lost money. Then they breeding medium size, not smaller size. They breed for medium size seed, but larger number of seeds per class. And so that's even right now they are trying to breeding medium sized seed, not the large seed anymore. So.
Yeah, I think everyone here who's worked with pennycress can say that we're not. I think larger seeds, even if it comes at a cost of reduced seed number, everyone will appreciate larger seeds because they are easier to handle. I mean, right now we're talking about seeds the size of a sesame seed. So anything that makes it easier for farmers to plant and harvest and transport and crush plants to handle, I think going for larger seed size, I don't think we're going to see a lot of negative effects just due to the benefits of seed handling.
Yeah, I agree with you. I'm an alfalfa breeder now. Debbie always mentioned we need to increase the seed size of alfalfa. So, excellent. Yeah. Thank you.
Yeah. Yep. Yeah, so there's a question in the chat from David, and it says, breeders have made tremendous gains in all crops. Can you make a prediction about what pennycress might look like in 10 years? Man, that's a hard question because I think we haven't really thought a whole lot. I think we're just beginning to think about if we're really truly going to always plant this in a relay cropping situation in Minnesota, if you know, are there structural changes? Are there specific maybe idiotypes we should aim for when we are breeding for that relay cropping system? And you know, I think that would help a lot. However, you know, I think about some of pennycress, when it tends to lodge in St. Paul, it's because that main stem cracks at the bottom. And so, you know, I think working with a plant physiologist to think about how we can modify the plant structure successfully would be really helpful in figuring out how pennycress might actually look in 10 years. Because I do think if we want to continue to relay crop this, we're gonna have to really think about the structure and how pennycress grows so that it facilitates harvesting over the top of soybeans. Now, if we're able to go to a true double cropping situation, I think what we'll start looking for is, you know, bigger, more vigorous plants that yield as much as they can. So that's my thoughts. Just having known pennycress in the very beginning of its domestication. So.
Yeah, one thing that's kind of motivated that question, you know, is that the Kernza story is kind of nice in that you can kind of see a linear sort of increase in like seed size and that over the years. And I'm just wondering if there's any trend that we can see yet for that in pennycress as far as selecting for traits and seeing, you know, is there a line that you can make on selections on making increases in seed size or other traits?
I mean, we can definitely make those selections, and we are starting to make those selections. Can we actually see the results of it? We're not really going to see significant results until we are only forward breeding. And we right now, because we're adding genetic diversity into our breeding population by adding in more wild lines, we're really not doing that much forward breeding. We've only and what I mean by forward breeding is when we're able to make only elite by elite crosses and very rarely elite by other crosses, or elite plus mutant crosses by elite plus mutants. I think that's when we're going to see a lot more significant changes.
Yeah, I'm wondering if the covercress folks here, Mark, I see is here. I wonder if Mark could make a comment about that, since I imagine he's done a fair bit of, maybe done some forward breeding.
Yeah, so we're, I mean, that's really been the basis of our whole program, is to get to that point as quickly as possible. And in fact, you know, we haven't been able to do up until this year, really elite by elite, say low fiber yellow phenotypes crossed together. But in fact, this year we have been able to do that, and we'll start to see those in nurseries and trials next fall. And I think I would very much agree with Katherine that, you know, I mean, and the reason we've done that, we've actually focused less on diversity and more on that, and it's because of our commercial situation and our funding situation, and that we need to demonstrate, you know, yield progress quickly first to get the funding, and then we'll worry about the diversity next. And I know, you know, obviously we talked a lot to Katherine about the breeding position here, and one of the focuses that I think when I step back next summer, one of the focuses for the new breeder will be to devise ways to incorporate new germplasm into our pipeline while still carrying forward that forward breeding mindset, because we do need to move yield forward quickly, even after we've commercialized the first cover crest.
Yeah, well, I'll be excited to see how that pans out in the future, and after we get the traits in and the breeders can actually do the thing that they do.
David, that's a good question. And as I started in Pennycress in 2013 with Kayla, and she grew out her materials, and as I walk the fields and see what Catherine's got out there, it's remarkable to me how little visible variation there is. I also run the spring wheat breeding program. There's a lot more variability in spring wheat, and we've been visible variability, and we've been breeding that for more than 100 years at this university. And David mentioned Kernza. Kernza is an outcrosser, and anybody can walk in the field and there's no two plants that look the same. There's so much genetic variability being an outcrosser. And the way I like to describe these breeding programs is that they're in their honeymoon phase, and we should be making some fairly fast gains at this point. We have in Kernza. What I see from the Pennycress data is these early crosses, they are performing better than their parents. So there is some genetic variation there, and we're able to take advantage of that. But going forward, I think the key will be to get these mutant traits in, but more wholesale genetic diversity is going to be needed to move it forward. And then we'll be, as the pipeline gets more full, we have more breeding material, we'll be throwing genomic selection at this too to try to speed progress that way.
Yeah, that was, it's funny how things change once you get into them, but actually in the first grants that I wrote, and this was, I don't know that I'd been here a year yet, was to use genomic selection in Pennycress. And then I got into my first field season here and said, oh, this isn't the time for genomic selection. One, we have to have plants that grow uniformly. Because that very first year, I could have made all of my selections for that year in the fall and never gone back to the field. So I think that's really, we do have to approach each of these crops differently. And I think we are now building the foundation through like Zenith's project and the crosses that I've made, and adding in the new genome. I think we're setting the stage to actually utilize genomic selection well in the future. There was another question that I think, there was a question about the TT8 mutant, and someone had asked me a direct message a little more about the background of TT8 and the oil profile. So I'll answer Tim's question briefly and then kind of let Ratan add in. And Tim's question was that he was intrigued that John's TT8 mutant performed so well in our trial. And can you tell us what genetic background it is in and speculate why it's done so well? And have we tried other TT mutants? And the answer is yes. We have tried other TT mutants, and we even had the Min106 version of TT8 in our trials. And we don't know why, but John's TT8 does a lot better than that one. I don't remember what John can add what genetic background that is, but for some reason that specific TT8 line really likes Minnesota. So
I'll say too that our TT8 has a lot of mutation drag in it, and so that's one of the advantages that John has with his CRISPR. Go ahead, John.
So that particular TT8, I think it was TT8 minus T is what you wrote. That's the
Yes.
ARB1. That's
Yeah.
the ARB1 mutant back or ARB1 breeding background. There's also the TT8 minus 2 base pair. That's the spring 3-2 background. I don't know if you've been planting that up there as well. I think as part of the I prefer strip plots, it's in there.
Yeah, I think we have it for sure this year. I don't think we had it last year in the I prefer. But John or Ratan, do you want to say what the oil profile of that TT8 line looks like?
So we're consistently seeing higher total oil, about 10% higher, but then that's sort of canceled out by the FA1 mutation, the lower Rusic, as Ratan was pointing out. That gives us about a 9% less oil. So it seems to be additive effect where we get just a little bit more oil with TT8 FAE double.
I mean, we see almost 10% increase in TT8 line versus the 106 line consistently.
So I had a question, general question to any of the breeders here. What are your thoughts on polyploid pennycress? Historically, based on other crops where you've gone from diploids to polyploid, do you think this has a chance of working in pennycress? Will it lengthen the crop, the time to maturity where it won't work perhaps, or that's not necessarily the case?
I can jump in. I mean, so David started the tetraploid pennycress program back in 2015 with Kirk Amundsen, and we've been propagating those lines. And what we've seen generally, those lines tend to mature a week later than 106. We do definitely see bigger seed size, bigger plant, vigorous plants. One thing we do notice is the plants are a little shorter than the 106 plants. So, I mean, it definitely pushes the maturity back. Others can add more to it.
Yeah. I can spill the beans there and just simply say too that just recently I'm working on making some tetraploid early flowering mutants to see if it will compensate for the lateness of the tetraploid business.
Do you think you're going to see a higher yield in those? The plants are a little bit bigger?
I think probably going back to what Katherine said, if we can just get something closed but have larger seeds, which is the kind of the hallmark. With the tetraploids, I would say we average around 1.6, where co-grown 106 is around 1.2, 1.3. And we're just touching the surface of that. We haven't done intercrosses between our tetraploid lines, and to see if we can get them to start thinking about becoming diploids, like in that we have a lot of, if you look at the seed pods, number of seeds per pod, it's way down. And it's because as an autotetraploid, there's reduced fertility.
I think, the big challenge with trying tetraploids is we already have a problem with genetic diversity, or we already have a limitation with genetic diversity right now. And we haven't necessarily gotten all the improvements that we want in pennycress. I wouldn't, as a breeder, I wouldn't really want to start playing too much with tetraploidy until I had what I truly considered a domesticated line. Just because, I mean, we can't, it would be challenging to make, much improvement with diploid by tetraploid crosses, right? So how do you keep improving, or do you create a tetraploid line for everything that you're generating through your diploid program?
Yeah, well,
I just think it's pretty early to, I think it's a great tool, but it's kind of like the genomic selection that the time wasn't right to use that. So the question will be, when do you figure out, or, what is the right time to use that method?
Yeah,
I
and
mean, just a simple question is, I mean, just can we improve the flowering time by combining it with an early flowering guy? So I'm not trying to make anything commercial here. I'm just exploring hypotheses.
yeah, definitely. But one thing I can comment on, like having tetraploids in the breeding program would be challenging because like just this few tetraploids that we've been propagating for last 7 generations, I would say. After 7 generation, now we see consistent pod fill, uniform seed size in the pods. So if you have to do that for every single line that we make a polyploid with, that's going to be like a long wait for each line. And then it's a, I don't think it'll be efficient in the near terms, but definitely maybe in the long term.
Yeah, certainly we can only hope Pennycress will be around 20 years from now.
Any other questions? We're almost an hour and a half here.
There's Don. I knew he would come on.
Don.
Yeah, well, I just want to say something to Katherine. I'm glad you took the risk in coming to Minnesota to be part of the Forever Green initiative. I knew you liked the philosophy behind the program. And as we discussed many years ago in terms of whether or not this program could give you the opportunity to develop your scientific base. And just to tell everyone, as I started putting these teams together 20 years ago, that was really what I worried about in developing teams around 15 new crops and bringing in young people and young faculty to be part of the program as to whether or not we would be able to give the opportunities, the scientific carrying weight around these crops. And what I figured out is that we can, right? You know, at least 5 or 6 of the graduate students and postdocs that have come into the program have now taken faculty positions around the world and also USDA positions. So that was one of the things I really, really worried about over the years. And I think, again, Katherine, you're a great example that the environment that you helped create here also helped develop the scientific base to recruit additional folks into the Forever Green initiative and around the development of these new crops. So you've made a major contribution to the development of new crops through all of your contributions to the Forever Green initiative and specifically to Pennycress. So we will always be, let's put it this way, I will always be indebted to you for what you've done for the program and helping build these relationships with faculty here and graduate students here on the St. Paul campus, as well as with the other components of the I Prefer program. And you provided great leadership. We're obviously sad to see you go, but we're also glad to see you go to this great new opportunity for you that you've been thinking about for many, many years. So I just wanted to express how thankful I am personally for what you've contributed and wish you the best in your career in Nebraska.
Thank you, Don. I really appreciate it. I know I've learned a lot. And one of the important, and this is a comment for the grad students or postdocs that are still on the call. What we do in the Forever Green initiative is the interaction, the relationships that we build, the cross-disciplinary work we do is very unique. And that's one of the reasons why I was excited to come here. And I will tell you that as I've interviewed, that skill set has gotten people very excited. And it's one of the things that, one of the reasons that I was hired at Nebraska versus some of the other candidates, because I have that unique relationship, cross-disciplinary research building skill set that I wouldn't have developed without the framework of Forever Green. And so I just really encourage everyone to continue to make that effort to build that skill set, because it's something that is of great interest to not just academia, but companies as now too. So utilize that and, take that message out to the places you go to. I hope to be able to do that at Nebraska.
Well, now that you'll still be working on winter barley, you can still be a Forever Green player.
It is true. It is very true.
Thanks again. Appreciate it. Love you.
Thank you.
Well,
I'll
thanks
miss
for all
you
your
all.
contributions, Catherine. We will be in touch over the next few weeks and years as we continue as colleagues working on wheat.
Yeah.
So looking forward to that.
Yep.
Yeah. Catherine,
Thank you, Zenith, as well.
I also want to say we enjoyed working with you very much, Catherine, at CoverCrest, and you'll be thoroughly missed by everyone on I Prefer. So please stay in touch. Come to our meetings.
I will.
Thank you.
Thank you, Catherine. Thanks for all the friendship we had for last 3.5 years. And we'll stay in touch even after that.
Yep. You all know, you'll all be able to find me. So.
Thanks, Catherine. We're gonna miss you big time, that's for sure.
You'll forget me eventually.
I don't think so.
Yeah. All right. Well, my realtor's been calling me, so I probably better return that call. So I will see you all some other time.